Thanks very much. Is this on? Yeah. Okay, so thanks very much, Afshan. It's a pleasure to be here. This is my fifth time in Malaysia, but first time at IMU. So I'm delighted to be an external examiner and seeing some of your wonderful students perform under the most stressful of situations, but in a really they're excellent individuals. So I'm delighted to be here. As mentioned, I work in Galway on the west coast of Ireland, which is one of your partner medical schools. So we welcome about 10 of your students to Galway every year. And so I've had lots of opportunities to engage with them in Ireland, but it's also nice now to come to see their birthplace, essentially. I'm going to talk today about the potential therapeutic use of mesenchymal stromal cells for treatment of diabetic complications. Just to begin, by way of a conflict of interest, I'm a founder, director, and equity holder in a company called Orbsen Therapeutics, which is a stem cell company. In the latter part of the presentation, I'll be referring to some of their technologies. So I'm an endocrinologist, and clinically I look after patients with endocrine disorders, the most common of which, of course, is diabetes. And the slide up here at the moment essentially shows the pandemic which is before us. And as I'm in Asia, if you point to the current numbers, predicted numbers, that's a 70% increase over the next, over a 20-year period. So of the projected approximately 600 million people with diabetes by 2030, 120 million of them will be in this region. So diabetes, in terms of a disorder of glucose metabolism and its association with hyperglycemia, the main problem of that over the long term is complications of diabetes, both macrovascular and microvascular. Macrovascular affecting the heart, the brain, the lower extremities, and microvascular predominantly the eyes, the kidneys, and the nerves. So if you go into a cardiology unit in Ireland, you'll see many of the patients there with coronary disease who have diabetes. Our dialysis units are occupied by many patients with diabetes-induced end-stage renal disease. And our ophthalmology clinics, and diabetes is the most common cause of blindness in the Western world. So these complications associated with a condition of increasing prevalence are going to have major impacts on the health system in years to come. So I'm interested in the use of a specific cell type, the mesenchymal stromal cell, as a potential therapy for these complications. And my presentation today will really focus on what might be considered experimental medicine. This is not a proven therapy. This is bringing together what might be viewed as an unmet medical need. Patients with diabetic kidney disease, diabetic eye disease, diabetic peripheral vascular disease, an unmet medical need brought together with the potential therapeutic use of a laboratory observation, and how do we transition from the laboratory observations into early clinical trial development. And that's really where mesenchymal stromal cells are at the moment. They're in that gap between preclinical work and early stage clinical trial development. And as I mentioned, the mesenchymal stromal cell, which we most commonly obtain from bone marrow, you can spin the aspirate and plate the cells on plastic. So these, unlike the hematopoietic stem cells, which do not stick to plastic, the MSCs do. And that's really the way of differentiating them from the marrow aspirate. And the cells grow over weeks to hundreds of millions. So you've got the capacity to take a marrow sample and grow up large numbers of mesenchymal stromal cells. They can be obtained from bone marrow, adipose tissue, umbilical cord, placenta, Wharton's jelly. So a number of different sources. We work a lot at the moment on bone marrow as a source, also adipose tissue. And we're moving more towards umbilical cord which is a readily available source and which in our country at least we discard as waste every day in our maternity units. Much easier to take one of those than have to take a marrow sample from a donor or a patient. Now, mesenchymal stromal cells, whether they're from bone marrow, adipose tissue, or umbilical cord, like all stem cells, have the ability to differentiate when they receive appropriate signals to certain cell types. And the mesenchymal stromal cell has tri-differentiation potential, can become adipose tissue, cartilage, and bone. So there are researchers in the bone field who want to use MSCs to convert to bone. In the cartilage field, they want to use MSCs to convert to cartilage in order to repair situations of defect in bone or cartilage. That's not why I'm interested in them in the context of diabetes. In fact, I don't want them to become bone or cartilage, but I'm interested in using MSCs because of their paracrine properties, their ability to secrete angiogenic factors, immunomodulatory factors, anti-inflammatory factors, and cytokines. So one may think of the mesenchymal stromal cell as a factory that generates therapeutic products. So if you're interested in treating ischemic disease of the lower extremities, the angiogenic factors may drive new vessel formation. If you're interested in diabetic kidney disease, where inflammation is a key pathogenic mechanism, then the anti-inflammatory factors and cytokines may have beneficial effects. So mesenchymal stromal cells are, have tri-differentiation potential. They're readily isolated and can be culture expanded due to their plastic adherence. They have a specific surface marker profile, and they're easily culture expanded. But they have this paracrine ability to secrete factors. So instead of using a single drug to target a pathway, in delivering cells, one would be delivering multiple factors. Now, I have this idea from the clinic, from the pre-clinic, from my laboratory work, that the use of MSCs, by virtue of their angiogenic or anti-inflammatory properties, can be used in these conditions. The way to progress from that scene to the clinical trial prior to clinical use is what might be referred to as the translational pathway. So one undertakes basic science, preclinical experiments in animal models, toxicology, and then progresses into phase 1 human studies, onto phase 2 and phase 3, which is the pivotal clinical trial, essentially to request market authorization for your product. So most of my presentation is going to dwell in this space, that between the laboratory and the phase 1 or early phase 2 clinical trial use. And I'm going to, my presentation is going to be in the context of the European regulatory framework. So the European Union has a whole series of directives which influence how we can do this translation. So before I came to this talk, I had an email from a man in Kuwait, who I don't know the man, but he said his wife has stage 4 chronic kidney disease. She heard of our work, and he wants us to give her stem cells for her stage 4 chronic kidney disease. Now, I've explained, that we can't do that. What we have is an experimental medicine, not a therapy. And in order to determine whether we can use it, we need to go through this path. And we get emails like that every day from patients with all sorts of diseases requesting stem cells for therapy. And we have to point out that you can go through a regulated clinical trial, but that's really the access to stem cell therapy at the moment. In MSCs, there are two approved products from MSCs now available for purchase on the market. One of them is for graft versus host disease, and the other is for fistulizing Crohn's disease. So those two products have gone through that whole path that I just defined, the preclinical laboratory, the early phase 1, phase 2, and pivotal phase 3. and they've received marketing authorization based on a review of all of that regulatory dossier for sale in the market. Now, the reason MSCs are useful in graft versus host disease is because of their immunomodulatory and anti-inflammatory properties. Probably likewise for fistulizing Crohn's. These were patients with Crohn's fistulae, perianal, that did not respond to standard therapy, were very troublesome for patients, and with an intrafistular injection of MSCs, the fistula closed more often than with placebo. And so that's the basis by which these two MSC products are now available for purchase for the treatment of those two conditions. So I'm an endocrinologist. As I said, I'm interested in diabetic macrovascular disease and diabetic microvascular disease. And the first one I'll talk about is macrovascular, focusing on the lower extremity. So critical limb ischemia, prevalence of 5 to 1,000 new cases per million population per year. In Ireland, that's 6,000 per year because we've got 6 million. In Malaysia, you have 32 million, so you can multiply that by another 5, presenting with probably 30,000 people presenting to their doctors with critical limb ischemia. Now, the treatment of choice is obviously to reverse the ischemia by bypass or stents, and about 2/3 you can, and about 1/3 you can't. And they end up then in the scenario where they require amputation. So I would say new treatments are urgently required. So I have this idea from the laboratory. MSCs make angiogenic factors. Could I take them from the patient's bone marrow, grow them, put them back into the leg, and see whether we could rescue or salvage the limbs? That's a hypothesis. That's an idea. It has to be applied through the translational process to see whether it's the case or not. So these are some of our preclinical data to support the fact that MSCs might be beneficial in this context. This is the secretome from bone marrow MSCs from non-diabetic and diabetic mice. Similar scenario with humans. And essentially every black dot is an individual angiogenic factor. So these cells make, when we culture them, all of these angiogenic factors. So this is part of the dossier that I will be preparing to present to my regulator to say that I would like to undertake a clinical trial of autologous bone marrow MSCs in patients with no option critical limb ischemia, those destined for amputation. But to do that, I have to show a preclinical rationale and a preclinical safety scenario. And I have to show I can grow the cells in a manner compliant with the regulatory requirements. So this is the first piece of evidence. The cells make lots of angiogenic factors. Second piece of evidence, when I put the media from the cells into an endothelial culture, they encourage tubule formation. So this might be an in vitro correlate of angiogenesis. And media from the MSCs do that, whereas negative controls don't. The third piece of evidence I'll present to you is if I take a DB mouse, which is this obese mouse down on the left, and I remove the femoral artery from the mouse, blood flow will fall. When I then inject cells back into that leg, mesenchymal stromal cells, blood flow recovery is improved relative to normal. So in a normal situation, there will be some improvement, some recovery, but that's augmented by the delivery of mesenchymal stromal cells. And also, when we do ambulatory scores of the animals, the higher the score, the worse the walking. It's improved also by MSC delivery. So I'm suggesting that I've got a disease with no good treatment option at the moment. I've got a cell which makes lots of angiogenic factors, which stimulates tubulogenesis in vitro, and which can promote vascular flow repair after removal of the femoral artery in an animal model. So that's my preclinical efficacy dossier, essentially. Now, in addition, there are 32 other published studies demonstrating the efficacy of MSCs in angiogenesis. So it's not just my laboratory has shown this. This has been shown by many laboratories. And this will be part of the investigator brochure that I present to the regulator as my application to undertake the human trial that I mentioned. The regulator is actually more interested in safety than in efficacy. They would love to see an efficacious therapy, of course, but they're really interested in safety. So stem cells, I mentioned they can become bone and cartilage. The regulator wants to know, will they become bone and cartilage? They're cells which can differentiate, can proliferate prior to differentiation. Regulator would want to know, can they become cancer? So we have to do a toxicology study. Now, toxicology in stem cell therapy is complicated because you have to use your final product in your tox study. And the final product is a human living cell. So you've got issues in putting that into animals. If you put it into a regular animal, there'll be a xenogenic response. So we have to use these specialized mice which have no immune system. And we put the bone marrow mesenchymal stromal cells, which we have grown under GMP conditions, into the animal to see is there any evidence of tumorigenesis or aberrant differentiation. And to save a lot of time, there isn't. So when we did this toxicology, we found no evidence of a safety signal. The third thing the regulator wants to know is where do the cells go after you inject them. So we designed an assay to look for human DNA in all the organs of the animal who had received this intramuscular injection. We're using human cells, so human ALU sequences. They're specific to humans. And we took all of the tissues and looked to see whether human DNA was present 3 months after the intramuscular injection. So the regulator wants some comfort to know that they're not going to be in the brain or the heart or the liver or the lung or places where you don't want them to be. And they weren't. Again, I won't go into this in detail, but this is the DNA assay. And it's seen to be present in calf and thigh in male and females. So we can find it at the site of injection 3 months later, but nowhere else. So that provides some level of comfort again in terms of safety. Now the other part, which for us was probably the most complicated part to get into a clinical trial, was that of cell manufacture. And Afshan mentioned I direct the Centre for Cell Manufacturing Ireland. So our university built a GMP facility, or good manufacturing practice, which just means it's a place which grows cells in a manner which is suitable to deliver the cells to humans after the production. Remember, human cells cannot be end-sterilized because they're living cells. You kill them if you end-sterilize them, so the process has to be sterile. And we built that facility, and then we had to show that we could grow cells. So we would take donor marrows, grow the cells. The regulator wanted us to do that three times. This is our license to show that we can do that. We procure marrow, 30 to 100 mils, in the operating theater, again insisted on by the regulator. You can't just take it anywhere. You take it in an operating. It has to be a hematologist, draws the marrow sample, and has to be procured under another license driven by EU regulatory requirements, a tissue procurement license. The marrow is then sent to our GMP facility, and it's grown in these flasks. So we spin the cells, as I said, plate them on plastic, and grow them in 40 of these flasks. Now this is, I'm going to refer in the second half of the talk to better and newer ways of growing cells. One of the problems with this is that it's very operator dependent. You have to have somebody changing the media, this red stuff. You got to change, so there's human intervention. So I'll talk later about trying to do this in a closed manner, which doesn't require any human intervention. These are the three, I'm not going to go through this slide in detail, but these are the 3 normal marrow runs that we had to do to get our license. So we take 3 normal humans, take their bone marrow, as I mentioned, grow up the cells, and show that it's sterile and that the chromosome is stable, and that we've got good viability in all the cells. And we did that, and we therefore had approval to manufacture the cells. And it was only at that point that we could then apply to do the clinical trial. And this is the clinical trial we propose to do. It's phase 1B, intramuscular autologous MSC delivery to critical limb ischemia patients with no option for revascularization. Phase 1B, open label, uncontrolled, non-randomized dose escalation study. These 3 doses, 3 patients at each dose. Primary endpoint is safety. Secondary endpoint is efficacy. And all of this is done under another set of regulations in a clinical research facility, which is specifically designed for undertaking clinical trials. So we submitted our preclinical efficacy, our toxicology, our biodistribution, our cell manufacturing data, and then we were approved to move on to recruit into this trial. And that preclinical phase probably took us maybe 3 to 5 years between construction of a GMP facility, making the cells, and getting all the preclinical data that I mentioned. So these are the patients we're recruiting. They're between 80 and 85. They have Rutherford class 4 or 5 CLI, no option for revascularization, should live more than 6 months, have no active malignancy, no inflow obstruction. So these are the patients who would present to their doctor with rest pain, gangrene, or ulcer. They would have an angiogram which would show no revascularization option, because if they did, they would go for revascularization. And then they would be essentially scheduled for amputation as in the current therapy paradigm. So they have the option to, with informed consent, enter this trial where a marrow sample would be taken. So here we recruit. We enroll after informed consent. We take the marrow. We send the marrow across to the GMP facility, grow the cells. And then the cells are released when we show they're sterile and when we show their karyotype is normal. And then they're delivered to the patient in the hospital, in the CRF, clinical research facility, into 20 intramuscular injections along the ischemic leg. So this is our clinical trial design. I'll tell you about some of the first patients that we treated with this paradigm, which will point out some of the challenges with an autologous approach. So this is the first patient, and he was a type 2 diabetic, 63, chronic kidney disease, came in with severe rest pain, and on opiates, and couldn't sleep at night, and had no option for revascularization. So he would have been offered an amputation. He enrolled in the trial, and it takes 3 months to grow the cells. So those of you who deal with patients with critical limb ischemia will realize that's a challenge. You know, the patient who's presenting with critical limb ischemia has to wait 3 months to get the cells grown up and released. And in fact, in this patient, in the 3 months, there was an advance in his disease. He had now developed gangrenous toes. And ultimately, although his pain resolved after the injection, he required an amputation 3 months after the injection. So that's the first patient, which is outlining some of the problems of an autologous approach. The second patient is an elderly man with a non-healing ulcer. He was treated, marrow was taken, cells were grown, he was treated, complete resolution of his pain, and we followed him up now for over a year, and the ulcer hasn't healed. And this is a low-dose cohort, 20 million, and the primary outcome of the study is safety. The third patient was an interesting man who was 72, type 2 diabetic. He was on dialysis, so he was on dialysis. He presented with severe rest pain and a non-healing ulcer on the second toe. He, after the treatment, has had full healing of the ulcer, and his pain is gone. So a year and a half after treatment now, he's pain-free without any ulcer. So that's the first maybe of the 3 patients who might show some signal of potential efficacy, with the caveat that this is not an efficacy study. It's unblinded, non-randomized, and non-placebo controlled. And the primary outcome to be assessed is safety. Now, because we had finished the first 3 patients, this gave us the opportunity to do something which has never been done before, which is to look at the MSC characteristics of an elderly, very sick population. We had grown many marrows from young, healthy people. We're very familiar with the characteristics, but not so much of older people, because you wouldn't normally take a marrow sample from a 75-year-old on dialysis. So what we did see is that the CLI patients have fewer cells, fewer mononuclear cells, and also they don't grow as well. So these are young, healthy cells, and these are the older patients. So maybe you might say, well, that's not surprising. Well, it may not be surprising, but it had never been demonstrated before that the marrow from an elderly patient, when you plate the MSCs, the cells, there are fewer of them, and they grow more slowly than young donors. This is the mean population doubling, showing again it takes a CLI patient almost 1.5 times as long as a young healthy to double their numbers. Another finding which we didn't predict nor expect was that some of the patients in whom we grew their cells had karyotypic abnormalities at the end of the culture expansion. So this is a trisomy 5 observation in the cells from one of our patients. Now that patient then could not be treated because this is a failed release. For us to release the cells to be administered to a patient, they have to be sterile, they have to be viable, they have the appropriate markers, and they have to be karyotypically normal. We have grown MSCs from young healthy donors for 15 years and never once seen a karyotypic abnormality. So again, this is maybe a signal about elderly people who are sick in terms of the stability of the cells on growth. So we moved on to the mid-dose cohort. I said the first dose was 20 million, the second is 40 million. And due to the slow growth rates, we decided to increase the marrow from 30 mils to 100 mils. This all required an amendment from our regulator. We grow 100 mils. We're planning to grow 100 mils of marrow in the theater, take it under conscious sedation. The fourth treated patient is very similar to the last patient I described, presented with rest pain, elderly man, CKD, not on dialysis, he did have a surgical option, but he was so sick the surgeons and anesthetists wouldn't operate. So he entered the trial, and his leg, this is at 30 days, we've now gone out to 90 days, his ulcer is healed and his pain is resolved. Again, single anecdote in the context of a safety study. So the safety results so far, which is the primary outcome of the study, we have found no safety signal, no safety problem. So none of the patients have had adverse events or serious adverse events related to the therapy. They have lots of serious adverse events, fewer serious adverse events, but this was related to their underlying disease. And that's adjudicated by me as PI, by an independent pharmacovigilance group, and an external pharmacovigilance group. The issue of causality and relationship between any happening and whether it could be related to the therapy or not. So to date, in terms of the efficacy, the rest pain is improving, and ankle brachial index, no change. So the summary in this phase 1B, which is an ongoing study, we have one more patient being treated this week, and one more in the next week or two, which will bring us up to the end of our mid-dose cohort. But to date, I would say that the therapy is safe with no evidence of treatment-related adverse events. There are karyotypic abnormalities. I showed you one. We found them in 2 others. So there were karyotypic abnormalities in 3 of the 9 patients that we've enrolled so far. We've had difficulty in expanding to required numbers. And although small numbers of patients, there's a trend towards improvement in pain, with 2 of the patients, the last 2 I mentioned, going from opiate dependence to no need for pain medications and no pain at all. So that's the end of the macrovascular part, and I'll just turn now in the last maybe 15 minutes or so to the microvascular work we're doing. We're not as advanced in our clinical trial enrollment, but we have 2 trials now open for recruitment for microvascular complications. I'm also going to reference in this part of the talk some technology developments. I showed you earlier we're using bone marrow as a source. We're talking about autologous, although I have concerns about autologous, as I mentioned. We don't sort the cells, so we just plate them on plastic. We grow them in flasks, and we use fetal bovine serum for culture. The American regulator won't allow cells grown in fetal bovine serum because it's a xenogenic product. The European regulator will, but probably will not in the near future. So we have to try and get away from fetal bovine serum to grow our cells. We're moving towards an umbilical cord source. We have a sorted cell product, which is more homogeneous and therefore more regulatorily compliant. We've developed a closed bioreactor system for cell manufacture. We're working towards xeno-free. We're not there yet. and we hope to have an allogeneic off-the-shelf product which is cryopreserved and ready for use. So I told you I'm a founder and director in a company called Orbsen, and Orbsen have a technology which sorts the cells based on CD362. This was driven by the FDA saying that these MSCs are a heterogeneous population, and the FDA are encouraging investigators to make the population more homogeneous. And one of the ways of doing that is to sort them based on some specific characteristic. And so they're looking for markers which may guide knowledge of mechanism of action, and the flexibility to improve product by adding additional markers during product development. So this is a kind of an edict or mandate from the FDA, the US regulator, to come up with a cell product which is more homogeneous than the plastic adherent method that I just mentioned. So OrbCell from Orbsen Therapeutics is a cell product which is sorted based on CD362, which is an antigen expressed on the surface of a fraction of the mesenchymal stromal cells. And we do this sort via MAX or FAX, and then expand in a closed automated bioreactor and have a fully closed automated manufacturing process, and then cryopreserve it so that it would be ready for delivery at the time of patient presentation, rather than needing to take the patient's product, grow it up, and then use it. And this is a graph of the purity, showing that the Orbsen cell is much purer based on the sort. Now, following the sort, it's then put on plastic adherents in order for it to grow. This shows that there are this is adipogenesis. I said the cells have tridifferentiation potential. There are very few adipocytes in the OrbCell product. This is suggesting that it's taking out impurities in terms of cell populations which have the capacity, for instance, to become fat. So having developed this technology, we applied to the European Union for a grant to explore the use of a CD362 sorted, closed expanded cell population in diabetic microvascular complications. And we were funded under a program called Red Star, Reducing Diabetic Damage Through Stromal Cell Administration. And this was in the first 24 months of Red Star. We looked at preclinical safety and efficacy, and in a manner I described in the CLI part of the presentation. And then the plan was to move on to a clinical trial based on those results in the second half. So these are the 6 complications: the kidneys, the eyes, the nerves, and then heart, bone, and ulcers were the other complications. So the cells were manufactured and then delivered to sites around Europe who had animal models of these complications to look for preclinical efficacy. Because if you don't have preclinical efficacy, then you're not translating. You have to show preclinical efficacy in a relevant animal model. So this is a diabetic ulcer model, believe it or not. This is the rabbit, alloxin-induced rabbit ear. You can do a punch biopsy and form an ulcer on the ear. And then you can apply the cells on a scaffold to that. So this is the design of the preclinical efficacy study, where we give alloxin, make the animal diabetic, wait 4 weeks, then induce the ulcer, and then put the cells on a scaffold. This is a topical delivery. And then look later, euthanize the animal, and look later for healing. And this bar graph just shows that the combination of the scaffold plus the CD362 sorted cells was most effective at closing the wounds relative to the other treatments. So that's the preclinical efficacy data, and we have histology to show angiogenesis as a potential mechanism of action. After that efficacy, we moved on to do a toxicology study. Toxicology must be done with your final product, and it must be done in a mode of delivery that you're going to use in your human study, which is topical. So in our toxicology study, we showed no evidence of aberrant differentiation, cancer formation, or any problems. And then we did a biodistribution study. So you're going to see a kind of a common theme in terms of if you want to translate, you have to do preclinical efficacy, preclinical toxicology, and biodistribution with your cell product. And each cell product will require new efficacy and toxicology. So in the first half of the talk, I presented a plastic adherent, bone marrow derived. I'm still on bone marrow derived, but now I'm sorting and so therefore it requires a new set of toxicology. So we had efficacy, toxicology, and biodistribution showing that topical application for neuropathic ulcers, so the target here is not ischemic ulcers, because for ischemia you have to restore blood flow. For neuropathic ulcers, they can be treated with local treatments. And we now have a phase 1b trial approved by the Danish regulator. So we're doing this trial in Copenhagen. The bone marrow has already been harvested in Galway. We've manufactured the cells on this occasion in the Netherlands in Leiden. And we manufacture the collagen in Scotland. These cells will be administered in the Red Star phase 1b trial at Steno, which is a diabetes center in Copenhagen. And that trial is now recruiting. And the patients who are being recruited into that trial have non-ischemic, non-healing neuropathic ulcers. If they had ischemia, they would need to go for an intramuscular delivery. But if they don't have ischemia and they have a neuropathic non-healing ulcer, they can enter this trial. And it's a similar design. It's a phase 1b, 9-patient dose escalation study. Now, we also looked at nephropathy in an animal model, and you remember, you're going to see efficacy, you're going to see toxicology, you're going to see biodistribution. So in this part, this is a group from Munich who are part of Red Star, and they have a model of diabetic nephropathy. This is the DB mouse who you also saw in my critical limb ischemia work. In the DB, they take out one kidney, and then by about 4 months, the other kidney has diabetic nephropathy. So we send the cells to Ackermanders, who's the PI in Munich, and he injects the cells IV to see whether they'll make anti-inflammatory factors that will protect the development of diabetic nephropathy. And up here, you see that the delivery of cells improves the GFR. So GFR increases after delivery of the cells. Proteinuria decreases. These are inflammatory cytokines measured in the kidney. So they reduce. And this is a morphology readout of the glomerular damage. And there's least damage in the CD362 IV delivered cells. So that's preclinical efficacy. And we then went on to do toxicology and biodistribution, showed no safety concern. Now this time it's IV we're going to deliver, so we have to do a new toxicology with IV. The first one, CLI, it's IM, we did IM tox. Topical, we did topical tox. And here we're doing IV tox. And this time it's a cell, it's a CD362 sorted bone marrow derived cell, but it's going to be allogeneic. So we're not proposing an autologous approach in this occasion. So we've just received funding from the European Union in the last 18 months to do Nephstrom. Nephstrom is stromal cells for diabetic nephropathy. And it's a follow-on from the Red Star positive preclinical results that I just described to you. This is going to be a multi-center placebo-controlled phase 1b/2a clinical trial done in 3 centers, actually 4 centers in Europe, including Galway. We're going to manufacture the cells in this occasion with a closed system, which I'll tell you about in a moment. The idea is to take 48 patients, randomize a quarter of them to placebo, and the other 3/4 to 3 escalating doses. This is the closed automated GMP manufacture system. It's made by a company called Terumo. And Terumo have cartridges in this system. This is the media that we previously would have had humans remove and replace. In this case, it's all done by the machine, and there's no human involvement. The cells are put in the cartridge, and the media washes are done automatically. So we had to get regulatory approval for this, and again show them that with multiple marrow aspirates that we could do this reproducibly, safely, non-infectively, with normal cardiology. And I'm not again going to go into detail. These are 6 separate marrows done by the consortium of GMP facilities we put together in Europe along with our one. And there's no evidence of sterility problems. Karyotype is normal. Now again, remember, this is going to be allogeneic, so these are young, healthy donors, unlike our CLI autologous donors. So Nephstrom will use this quantum to grow the cells in 3 GMP facilities. The first part of the process will happen in Leiden in the Netherlands. They'll pass on to, this is in Galway, Birmingham here, and Bergamo in Italy, and they'll grow the cells and distribute them to patients at each site. So Nephstrom will deliver a clinical trial, which is a combined safety efficacy. So on this, we're going to have placebo, double blinding, randomization, but we're going to start at a low dose and gradually increase the doses. Be placebo controlled. Primary objective will be safety. Secondary objective will be efficacy, which will be essentially to look at the GFR and what happens to the GFR. The people enrolled in the trial, the trial partners are in Galway, Birmingham, Belfast, and Bergamo in Italy. The subjects we'll recruit, 48 type 2s with 5 or more years. They have to have this degree of proteinuria. They have to have a GFR between 30 and 50, and they have to have had a decline in the GFR over the previous years. So there's very strict inclusion criteria, because we want to take patients who are destined for end-stage renal disease and see if we can alter the slope of the decline, which we will have 3 years of evidence of before the intervention. So this, this trial is currently open for recruitment across the three countries, 4 centers. The last part of the presentation is just going to mention 2 other complications that we've done the preclinical work, but we haven't yet got to the clinical trial stage. And this is neuropathy. Male Wistar rats given streptozocin. They're rendered diabetic. And this work is done in Red Star by my colleague in Porto in Portugal, Isora Tavares. So she's a neuropathist. Her work revolves around diabetic neuropathy, and she is a model of both painful and painless neuropathy in these Wistar rats. We send her the cells, and she doesn't, and we send her placebo. She doesn't know which is which, and she injects and looks at the effect on neuropathy in this animal model. And I'm not going to go into this in detail, but these are control animals down here in terms of a score for thermal hypoalgesia. So she is an experiment where there's a hot plate and the animal will withdraw the limb from the hot plate, but they'll withdraw it more slowly if they have hypoalgesia, if they have neuropathy. And this is the control. Diabetic animals have a much worse score on this hypoalgesia. And then when we inject intravenously in a dose increasing manner, we see that one dose, the mid dose, brings the cells back down to a non-diabetic range essentially. So that's preclinical efficacy data, and she has it in a number of other neuropathy readouts that I won't go into in detail in the interest of time. This is another one of the neuropathy readouts. It's its sensitivity to pain. These are the controls, and the streptozocin diabetics are down here. And when you inject the cells, it's again the mid dose brings the score back up towards the non-diabetic range. So 2 separate neuropathy readouts of IV delivery of a CD362 sorted bone marrow cell, and it's done by an independent group who don't know what they're actually injecting until we unblind after the analysis. The last preclinical set of data is in diabetic retinopathy, and this is done by under Red Star again by our colleagues in Belfast, Alan Stitt. He's a diabetic retinopathist, so he works on diabetic retinopathy, and he's got animal models. One of the key features of diabetic retinopathy early in the course is acellular capillaries. So he has this streptozocin model of being able to take the eye and look at acellular capillaries. And here, if you have this is a citrate PBS, number of acellular capillaries. This is a diabetic. So you can see the number of acellular capillaries increases with the induction of diabetes. And this is after the injection of CD362 positive cells. So there's a return to the non-diabetic range. So the IV delivery in this retinopathy model improves the morphology. And this is another readout of retinal ganglion cells. So they reduce with diabetes, and after the IV delivery of CD362 sorted cells, they go back up to the non-diabetic range. In terms of another technology development, I talked about sorting. All the work I've presented has been done with MAX sorting. It's a magnetic bead sorting. We've moved on to develop under Red Star a new technology with a company called OWL. They developed this tabletop GMP compliant fax sorter, so we can put the marrow sample in and the sorting will happen under fax. The development of this has allowed OWL to be acquired by Miltenyi, one of the biggest companies in the world who make sorters. So we will be moving away from a MAX and towards a fax system of cell sorting. So our cell manufacturing goal ultimately is to have an allogeneic and off-the-shelf product. We'd like it to be well characterized and be a pure starting material because the regulator is going to insist on that. We want an umbilical cord source. I've shown you no umbilical cord data. We have a whole program which is working on developing umbilical cord as a source for CD362 sorted MSCs. We'd like the cell population to be fax sorted in a GMP compliant manner. We want a closed system of expansion with xeno-free products. Again, I'm still working with FPS in all I've described to you. And we have another EU grant called AutoStem, which is a €6 million program to develop robotics and remove humans from any part of the process of cell manufacture. So to summarize, I've shown you preclinical efficacy, toxicology, biosafe biodistribution, and this is available for diabetic microvascular and macrovascular complications. In the macrovascular space, using bone marrow unsorted cells in an autologous manner, we've progressed into phase 1b clinical trials. And in the microvascular, all of the preclinical data sets has now resulted in approval of 2 human trials in nephropathy and diabetic neuropathic ulcers. And we have animal data for the other 2 complications, which we will progress in the coming years. So clinical trials are underway. We're working in parallel with all of this process to advance cell manufacturing technology using fax sorting of cells, using a pure cell population with CD362 sorting, and using a closed system for cell manufacture. This is our newly built clinical research facility funded by one of our government funding agencies, the Health Research Board. And this is the team essentially involved in manufacturing, in acquiring the marrow, in manufacturing the cells, and in the clinical research facility. These are my colleagues who I'd like to acknowledge, vascular surgeons, these 2, nephrologists, and some of the PhD students and postdocs in the lab. This is the west of Ireland, so if you visit us, this is our back door in Connemara, and this is the Atlantic Ocean, and across that direction is Boston. And these are the funding agencies. So thanks for your attention, and if you have any questions, I'd be happy to try and deal with them in the context of what was a lecture really which focused on the translational process, how to get from basic laboratory discovery through preclinical efficacy onto toxicology and biodistribution into early phase 1b, 2a clinical trials. Whether any of these products will make it to market authorization, as has occurred in GVHD and in fistulizing Crohn's, remains to be seen. Thank you. Yeah, no, I think that's an excellent question. I mean, I'll reiterate again, I should mention actually for the recording that the question is about the heterogeneous clinical outcome in the 4 patients who've received the cell product, and is there any way we can predict what the outcome is going to be? And that's a crucial question. You can't really from a phase 1b study. So a phase 1b study is about safety and feasibility. So you're going to the regulator with a request not to study a therapy as such, but to study an approach, and you're looking for safety in humans, which you've already shown in animals, to allow you move on to the phase 2, which will have controls, and then on to the pivotal phase 3. In every disease we treat, I think, with standard therapies, there are responders and there are non-responders. And I don't think we have a good handle, even in current conventional medical practice, on who's likely to respond versus not. Pharmacogenomics are going to hopefully add to that, and we're going to be much more precise at deciding who, when they get a statin, will have a good LDL drop versus limited LDL drop, or who will get myalgia with a statin. So I don't think it's a whole different question to what would apply to standard therapeutics, but in stem cell therapy, we're at a very early stage in the translational process. At phase 1b, you're not going to be able to determine that, but you will have to design it into future larger trials and see can you predict. I couldn't comment in any of these 4 patients. 2 of the 4 patients had resolution of pain and complete healing of ulcers. If I do nothing to patients, some will also have better pain and healing of ulcers. So the ones who are scheduled for amputation, those who actually don't get it for whatever reason, some of them actually get better. So I'm very circumspect in suggesting that what I've shown you in those 2 is efficacy. It's not proof in any way of efficacy. All I can tell you from my phase 1b trial is what it's designed to do, which is that it's feasible to take cells, grow them up, with the caveat that growth rates are not as good as young normals, with the caveat that 1/3 of our patients have developed karyotypic abnormalities, and that we can put the cells into patients in an autologous manner, in a safe manner. There is no treatment-related adverse effects. But your question as to efficacy, and more importantly, how to predict efficacy in a population, I don't think that's even well known for conventional medicines right now. And developments and advances in areas such as pharmacogenomics, where you genomically profile somebody, which will suggest either toxicology or efficacy, we're not at that point yet. I think you're asking me about FBS anyway. So in the first part, in fact, in all of what we've done so far that I presented to you, we used fetal bovine serum to grow the cells. Yeah. 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 That's a fantastic question, and one that keeps me awake at night. There are a few things that keep me awake at night. That's one of them. Each manufacturing development we make, is it going to make us go all the way back to the beginning? At what point is a change in the manufacturing process of such substance that you have a new product that requires, as you said, repeating the phase 1b, but maybe even going back to repeat preclinical efficacy, etc. It's a regulatory issue, and we engage with our regulators substantially, frequently. Our toxicology program is very much driven by what they want. I think, and we now have a cocktail of growth factors that will grow cells as well as FBS, and they're all human, so there's no xenogenic component. I expect the regulator will want us to go back and show equivalency of the cells grown under growth factors and under FBS. What I'd hope they would allow us do is show equivalency in some animal assays, and then accept that if we've done a phase 1b that we could progress straight to a phase 2A. But your question is right on the button in terms of manufacturing developments and what it does to your product. Drug manufacturers don't need to deal with this, because drug manufacturers make the drug as a chemical in a specified way. Cell manufacturers, because it's such an early phase of the development of cell manufacture, we're all trying to use different and better sources, different and better methodologies, and I hope one day to have one product like the pharmaceutical industry that will be used for every indication thereafter, so that I don't have to answer the very relevant question that you've raised, which is, do you need to keep going back again when you change your manufacturing process? So thank you.
One of the slides you mentioned about the mesenchymal stromal cells having a normal phenotype, screen for microplasma. Is there a particular reason why they're doing it? Is it a requirement of the regulation?
The cardiology is an absolute requirement, as is mycoplasma. Interestingly, with the cardiology, when I met our regulator initially, I suggested to the regulator that we don't do cardiology, because I said I have loads of data showing no karyotypic abnormalities. In the literature out there are rare individual cases with far passage cells of karyotypic abnormalities. So I tried to convince the regulator that I didn't have to do it, because it's another delay in the autologous approach, because it takes a while to get that test done. And the regulator said, no, you have to do it. And the regulator was absolutely right. Because if we hadn't done it, all the other release factors, mycoplasma, sterility and such, we would have gone on to inject the cells. Now, maybe it wouldn't have caused any problems, because what's our concern about karyotypic abnormalities? The concern is that you could have an unstable chromosome which might predispose to cancer. We've taken these karyotypically abnormal cells and we've done lots of experiments on them, and they don't form cancers. So it may be that it wouldn't have been relevant anyway, but we can't say that for certain. And the regulator was correct in this was the first stem cell trial ever in Ireland, so the regulator was dealing with something that had never been done before. So they took an ultra cautious approach. The only reason I was suggesting not doing cardiology was not, to be a cowboy and do things quickly, but because of the autologous nature of the trial, I knew these patients were going to progress with their disease over a 3-month period. But yes, there are regulatory requirements. Do we what? Sorry, the air conditioner is on, so I'm hearing everything about the paracrine factors, but are you saying, do we measure them, or do we profile them? Yes. Thanks very much. And, we do it by both angioarray, as I showed you in that one animal experiment. We do it by multiplex ELISAs, so we measure VEGF and FGF and by ELISAs, and it's pretty constant from one normal donor to another what the profile is. It differs from bone marrow to umbilical cord to adipose tissue, which again goes back to the earlier point of, if you go change source, you have to go back again. So yes, we do profile them. We know what the factors are, and we know that it's pretty consistent in normal donors. We haven't yet looked at diseased people and whether the profile changes. And there are some people who said, why not just don't use cells, use the conditioned media, grow the cells and take the stuff on top of them. In animal experiments, if you do that, you get as good an effect as you do with the cells. But it'll be almost impossible to get regulatory approval to do that, because your characterization of your product is going to be challenging, because the factors, you'd have to measure them all. And so what people are working on now is the use of exosomes. So MSCs secrete all those factors, but they also secrete exosomes, and the exosomes contain microRNAs and genes and proteins. And so the reason people want to get away from cells is you don't have to worry about malignancy, or you don't have to worry about differentiation problems. If you could use exosomes, or if you could just use the secretome, you would avoid those problems. But regulatorily, It'll be difficult to do paracrine secretome. Exosomes are on the way. There's a fair bit of evidence now that exosomes are as effective as the whole cell. So there's a lot of companies working on trying to standardize exosome manufacture, and you can grow 100 million cells, and each cell will secrete 100 million exosomes, so you get a vast number of exosomes. So yes, we do profile, and the question you ask, I think, is very relevant even in the context of, do you need the cells? You need them if you're doing a differentiation path, if you want to make bone, if you want to make cartilage, but we're trying to use them as factories, as cell factories. Could you not just use the product of the factories, or could you not just use part of the factories that isn't the whole cell, the exosomes? The exosomes being very actively explored, the secretome less so. Yeah.
So you've not tried to give a second dose?
Yes.
To see whether...
That's a good question, Professor Ramu, as well. And yes, we've only done one dose. And could we do a second dose? Yeah, in the autologous context, you have even less concerns about allogenicity. If you're doing allogeneic donors, do you use a different donor for the second dose or the same donor? And so all of that is, we have 3 immunologists who work in Remedy whose whole rationale is to try and understand that question. Can you repeatedly dose?
Because what I understand from literature, MSCs are not immunogenic.
They're hypoimmunogenic, yes. But we've shown in many publications now that they do induce an antibody response. They do induce a T-cell response, although on some of our re-administration experiments in animals, the T-cell response is actually T regulatory cells, which are hypo-responsive. So excellent question, big area of scientific exploration in the context of allogeneic MSC transplantation. In the beginning, it was thought that they were just naive, null, and not prone, but they do. They induce some immune response.
No, because I work with hematopoietic stem cell. We make dendritic cells. Yeah. And we give, the more dosage you give, we're seeing a better response. That's why I asked the question.
Yeah. No, I would love to know the dose, the frequency, and such, but all to be explored over the next, and maybe your medical students and other medical students. It's not me, I'm the past, they're the future, and maybe they'll figure out those questions around exosomes, allogenicity, immunologic issues, and which indications it works in and which it doesn't work in, because it's not going to work in all.
Just, I would like to know why you used intramuscular injection for diabetic septic food and for diabetic nephropathy and others retinopathy. You used intravenous.
IV, for retinopathy and nephropathy, we used IV. For critical limb ischemia, we use intramuscular. For non-healing neuropathic ulcers, we use topical. So, and you raise a very good question. It's almost like the reviewers of my grants are in the audience, because every single question I've been asked come up in reviews. Why do you not do intra-arterial, for instance, in critical limb ischemia? We did intra-arterial in a pig and found quite a bit of microvascular occlusion after intra-arterial bolus delivery of large, like hundreds of millions of MSCs. Now we reversed it with IV heparinization, so we're nervous about intra-arterial delivery of bolus MSCs because of the possibility of clumping and microvascular occlusion in an already ischemic leg. So that's why we did it intramuscularly, but we'd like to do it intra-arterially because you can do it through a device. You don't need the 20 injections. And then the topical is because with neuropathy, the flow is fine. You just want to get the factors to the side of the ulcer. And the IV for nephropathy is because we thought that it's going to be a systemic anti-inflammatory response, that the cells are going to make the factors and may help the nephropathy. With the eye, you could do it intraocular, or you could do it IV. We tried it IV, not expecting that it would work. We thought we would need to go intra, a bit like the Avastin injections nowadays and the new treatments for diabetic eye disease. But your route of administration, just like dose, frequency, source, autologous versus allogeneic, they're all the questions that kind of capture the minds of people in the translational cell therapy space. So I think it's an excellent question, again, as to why you choose any specific route for any specific indication. So thank you.